welcome to biol4u.com once again. Today I am going to explain glycolysis, or it is called EMP pathway. So glycolysis, it is a common pathway for both aerobic and anaerobic organism. Okay, so both aerobic organism and anaerobic organism, that means the organism which living in presence of the air, that is a aerobic organism, and also it is a pathway for the anaerobic organism, aerobic and anaerobic organism. It is a common pathway. Means glycolysis taken place in both aerobic and anaerobic organism. Okay, and this is the first phase of the energy yielding process in the cell, or in the respiration. This is the first process, and that is common to both aerobic and anaerobic organism. The word glycolysis, glyco means carbohydrate, glyco. So the word glyco means actually it is known as the sugar. Okay, and lysis means splitting. Lysis. The word lysis means splitting. So what is a glycolysis? Glycolysis means splitting of the sugar. Okay, so it is known as the splitting. So splitting of the sugar is known as the glycolysis. That is the word meaning glycolysis. Okay, the scheme of the glycolysis means the entire scheme of the glycolysis was given by three scientists. That is the Embden. The first scientist is the Embden. Along with the Embden, Meyerhof, the another scientist is the Meyerhof, and the third scientist is the Parnas. So this pathway is known as the EMP pathway. To respect these three scientists, the term glycolysis also known as the EMP pathway. So the Embden, Meyerhof and Parnas pathway. So in the anaerobic organism, it is the only process. I told it took place both in the aerobic and anaerobic organisms. So in the case of the anaerobic organism, means anaerobic bacteria or anaerobic organism, this is the only process in respiration. Anaerobic organism, it is the only process. Anaerobic organism, this is the only process of the respiration. Only process of respiration means the glycolysis cannot proceed ahead. So that is, it is only process of the respiration. So in anaerobic organism, this is the only process of respiration. Means the glycolysis cannot proceed ahead in the case of the anaerobic organism. In anaerobic organism, this is the only process of respiration, and the energy yielding by anaerobic organism only through the glycolysis, only process of respiration. But in the case of the aerobic organism, the condition is different, that we will study later. So hence it is a common pathway. I already told it is a common pathway for the both aerobic and anaerobic organism. And this process has taken place in the cytoplasm of the cell, not in the mitochondria. It has taken place in the cytoplasm. So, and this reaction, glycolysis, is independent of oxygen. It is not dependent on the oxygen. Independent of oxygen. Hence, it is a common pathway for both aerobic and anaerobic organism. So in this process, what happens? So during the glycolysis, glucose undergoing oxidation. Glucose. And this oxidation is not complete, it is a partial oxidation. So glucose undergoing partial oxidation, not a complete, partial oxidation. And by the partial oxidation, it produces two molecules of the pyruvic acid. Pyruvic acid, that is the final product of the glycolysis. Okay. And in the case of the plant, this anaerobic respiration, in the case of the plant, in this anaerobic respiration, the substrate used is actually sucrose. So this sucrose must be first converted into simple sugar, monosaccharide, that is the glucose. Sucrose is actually disaccharide. It converted into monosaccharide, that is the glucose and glucose plus fructose. So first it must, in the case of the plant, it must convert it into monosaccharide, that is glucose and fructose. This process is taken place with the help of the enzyme that is known as the invertase enzyme. So in the presence of the invertase enzyme, in the case of the plant, sucrose converting into glucose and fructose. In the case of the animal, the glucose already present there, and this glucose or fructose is undergoing glycolysis and that converting into 2 molecules of the pyruvic acid. So let us analyze the different process taken place in the glycolysis. You know, in the glycolysis there are 10 stages, 10 steps present in the glycolysis, 10 enzymatic chemical reaction present in the glycolysis. Out of these 10 steps, the step 1 to 5 is known as the preparative phase. Here preparing something with the utilization of the ATP also. Preparing something, so it is known as a preparative phase. And at the same time, the step 6 to 10 is known as the payoff phase. Payoff means this is the preparative phase. It is preparing something, so it is known as a preparative phase. And during the preparative phase, it utilizes some ATP, and that ATP is giving back during the 6th to 10th reaction. So it is known as a payoff reaction, and 1 to 5 is known as the preparative phase. So like that, 2 phases, and one more phase, that is there is a cleavage phase also, and that we will study when we analyzing the different steps individually. Okay, so let us study the 10 different steps that involved in the glycolysis. So let us study the different steps involved in the glycolysis. Okay, so the first substrate for the glycolysis is the glucose. So in the step number 1, this glucose undergoing phosphorylation and converting into glucose 6-phosphate. So this is an enzymatic reaction, and this reaction requiring an enzyme, that is the hexokinase enzyme. Okay, hexokinase enzyme. This hexokinase enzyme requiring a cofactor, that is the magnesium. So it is requiring the magnesium ions. So first glucose converting into glucose 6-phosphate, and this glucose 6-phosphate converting into fructose 6-phosphate. It is an isomer. Fructose 6-phosphate, this is an isomer. And here also an enzyme is requiring, that enzyme is the phosphoglucoisomerase. The enzyme is phosphoglucoisomerase. So with the help of this phosphoglucoisomerase enzyme, the glucose 6-phosphate converting into fructose 6-phosphate. Okay, and here 1 ATP is requiring. So 1 ATP is undergoing hydrolysis and it converting into ADP and inorganic phosphate, and with the release of phosphate group. This phosphate group is utilized for the phosphorylation of the glucose. So glucose converting into glucose 6-phosphate, and glucose 6-phosphate isomerized into fructose 6-phosphate. Then say the fructose 6-phosphate is again phosphorylated 6-phosphate, again phosphorylated and converted into fructose 1,6-diphosphate. Fructose 1,6-biphosphate or diphosphate is producing. So here see again 1 ATP is utilized, and this ATP is converting into ADP plus inorganic phosphate, and this phosphate is adding to the fructose 6-phosphate that is converting into fructose 1,6-diphosphate. And here also an enzyme is utilizing, that enzyme is the phosphofructokinase. Okay, that enzyme is utilized here. This also using magnesium as the cofactor. Okay, and then next this is actually the preparative phase. So this phase I already explained, that is the preparative phase. In the preparative phase 2 ATP molecule is used. Then the next stage, that is the cleavage. The fructose 1,6-diphosphate is broken into 2 molecules. These 3 glyceraldehyde phosphate, 1 molecule. Another molecule is the dihydroxyacetone phosphate. Dihydroxyacetone phosphate. So here fructose 1,6-diphosphate split into glyceraldehyde phosphate and dihydroxyacetone phosphate. And this reaction is actually taken place with the help of the enzyme that is known as the aldolase. So enzyme participating here is the aldolase enzyme. Okay, so this stage is known as the splitting stage, or it is known as the cleavage stage. So that is the cleavage. So I told you there are 3 stages. The first stage is the preparative phase. So this is the preparative phase. The preparative phase prepared prepared fructose 1,6-diphosphate that is split in the presence of the aldolase into glyceraldehyde phosphate and dihydroxyacetone phosphate. This stage is known as the cleavage stage. Then see here, these two molecules is undergoing isomerization. So here the glyceraldehyde phosphate converting into dihydroxyacetone phosphate and dihydroxyacetone phosphate converting into glyceraldehyde phosphate. So this is an isomerization reaction is taken place here, and this isomerization reaction is catalyzed by triose phosphate isomerase. So this reaction catalyzed by triose phosphate isomerase. So here reaction is isomerase. So it is isomerase enzyme, triose phosphate isomerase. So this reaction converting this glucose into two molecules of the 3-carbon compound, that is the glyceraldehyde phosphate and the dihydroxyacetone phosphate, and this interconvert with each other with the help of the enzyme triose phosphate isomerase. Okay, so then we will study the payoff phase, means next 6 stages, 6th to 10, that is the payoff phase. Okay, see here total 5 steps completed, and by this 5 step it produced 3 carbon compound, that is the glyceraldehyde phosphate and dihydroxyacetone phosphate. Then hereafter only glyceraldehyde phosphate is proceeding, means one glyceraldehyde phosphate entering to the next stage, then hydroxyacetone phosphate converting into glyceraldehyde phosphate, and that also coming downward. So hereafter each molecule is 2. So that is the reason why glucose converting into 2 pyruvic acid. So from this point onward each molecule is twice, because 3 carbon compound that entering to the next stage. So finally 2 pyruvic acid molecule we will get. Okay, so let us see the next payoff phase, how it operated. So now the cell containing 3 carbon compound, that is the glyceraldehyde phosphate. One molecule is the glyceraldehyde phosphate and another one is the dihydroxyacetone phosphate. These molecules interconvert, so it is known as the isomers, and here an isomerase enzyme is participating, that is the triose phosphate isomerase. Then glyceraldehyde phosphate entering to the next stage. So what happens here, this glyceraldehyde phosphate is undergoing phosphorylation and it converting into 2 molecules of 1,3 by phosphoglycerate, So here you can see one phosphate group is adding to this molecule. So one phosphate group must be given to this reaction, and this phosphate group is taken from H3PO4, not from ATP, but phosphate is taken from H3PO4, and this type of the phosphorylation, so that producing adding one phosphate group to the glyceraldehyde phosphate, that converting into 1,3 by phosphoglycerate, glyceric acid, and here a reducing agent is forming, that is the NAD, nicotinamide adenine dinucleotide, that converting into NADH, because a hydrogen is releasing at this reaction. Okay, so here the hydrogen is releasing and phosphate group is adding. So this reaction is catalyzed by a particular enzyme that is called glyceraldehyde phosphate dehydrogenase. Glyceraldehyde phosphate dehydrogenase, because the reaction dehydrogenase reaction taken place, so dehydrogenase enzyme is requiring. At the same time a phosphate group is adding, so it is called glyceraldehyde phosphate dehydrogenase. Glyceraldehyde dehydrogenase, and also phosphate group is adding, so it is called glyceraldehyde phosphate dehydrogenase enzyme. The main reaction is the hydrogen ion is releasing, and this hydrogen ion is adding to the NAD and producing the high energy molecule, that is the NADH, and this utilized later for synthesis of the ATP in the case of the electron transport system. So this reaction is actually the hydrogen is releasing, hence it is known as the enzyme is known as the glyceraldehyde phosphate dehydrogenase enzyme using, and 1,3 biphosphoglycerate is synthesizing. Okay, and then this 1,3 biphosphoglycerate undergoing dephosphorylation, and it produces 3-phosphoglycerate. it is producing 3-phosphoglycerate. 3-phosphoglycerate. Okay. So here one phosphate group is removing and that phosphate group is adding to the ADP and it produces one molecule of the ATP. And this type of the ATP formation, it has taken place in the substrate level. So this is the substrate level phosphorylation. Substrate level phosphorylation. So one molecule ATP is produced and the 1,3-biphosphogycerate converting into 3-phosphoglyceric acid. It is converting into 3-phosphoglyceric acid. And the enzyme utilized here is the phosphoglucokinase. The enzyme utilized here is the phosphoglycerokinase. Glycerokinase enzyme is utilized here. Okay, and this also wherever the kinase magnesium ions using as a cofactor, magnesium is utilized. So this 3-phosphoglycerate then converting into 2-phosphoglycerate. 2-phosphoglycerate. So this is an isomer. 2-phosphoglycerate. Okay, it is converting into 2-phosphoglycerate. So this is an isomerization. So here an isomerase enzyme is using, that is known as the phosphoglyceromutase. Phosphoglyceromutase enzyme is utilized here for this isomerization reaction. Then 2-phosphoglycerate is converting into phosphoenol pyruvic acid. Phosphoenol pyruvic acid. Phosphoenol pyruvic acid is producing. Okay, and this reaction involved the removal of the water molecule H2O. One water molecule is releasing from the 2-phosphoglycerate that is converting into phosphoenol pyruvic acid. Okay, and the enzyme utilized here is known as the enolase enzyme. Enolase enzyme. Okay, and this phosphoenol pyruvic acid undergoing dephosphorylation. So here one phosphate group is releasing and this phosphate group utilized for the synthesis of the ATP from the ADP. So ADP converting into ATP. So at two places ATP formation taken place and finally this converting into the product that is the pyruvic acid. So here pyruvic acid is forming. Pyruvic acid. So I told from this stage onward each molecule is 2. So each one is 2. So here finally the pyruvic acid also 2. 2 pyruvic acid will form here. So that is the end product of the glycolysis. Okay. So let us see this stage very clearly with the help of the diagram. Okay, the chart that is showing the different phases of the stages of the glycolysis. See here the first stage that is the glucose 6-phosphate. So this is the first stage. Here one ATP molecule is utilized and the enzyme hexokinase is acting and here this enzyme requiring the magnesium as the cofactor. Okay, magnesium cofactor is requiring here and glucose 6-phosphate converting into fructose 6-phosphate. Okay, here isomerization taken place. So phosphoglucoisomerase enzyme is acting here. Okay, then fructose 6-phosphate converting into fructose 1,6-diphosphate. So a phosphate group is adding to the fructose 6-phosphate. So one more ATP is utilizing here and here the enzyme phosphofructokinase. So this also using magnesium as the cofactor. Okay, cofactor is using and fructose 1,6-diphosphate then split, that is a cleavage stage, into glyceraldehyde phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by the enzyme aldolase. Okay, and here this undergoing isomerization. So here an isomerase triose phosphate isomerase enzyme is utilizing here. Okay, then glyceraldehyde phosphate converting into 1,3-diphospho or the biphosphogyceric acid and here the enzyme glyceraldehyde phosphate dehydrogenase is utilizing here and here again a phosphate group is adding and this phosphate group is not providing by the ATP, it is providing by the H3PO4 but at the same time NADH is forming. Actually from this stage onward 2 molecules producing. So 2 molecule 1,3-biphosphogyceric acid and 2 NADH is forming here. Okay, so this 1,3-biphosphogyceric acid converting into 3-phosphoglyceric acid. So here a phosphate group is releasing. This phosphate group utilized for the synthesis of the ATP molecule. So this type of the ATP formation is not taken place through the electron transport system, and hence it is known as the substrate level phosphorylation. So this phosphorylation is the substrate level phosphorylation. So this is the substrate level phosphorylation. Okay. And here the enzyme phosphoglycerate kinase is using, again magnesium is requiring as a cofactor here, magnesium ions. Okay, then 3-phosphoglycerate undergoing isomerization, and the position of the phosphate changing to second, and 2-phosphoglycerate is synthesizing, and here the enzyme utilized is the mutase. So this is a type of the isomerization, that is a phosphoglycerate mutase enzyme is utilizing here, and 2-phosphoglycerate converting into phosphoenol pyruvic acid with the help of the enzyme enolase. And this phosphoenol pyruvic acid under the dephosphorylation, that means the phosphate group is releasing that helping the formation of the ATP. Here also ATP is not forming through the electron transport system, and hence this type of the phosphorylation is known as the substrate level phosphorylation. So at two places the ATP is formed, and this ATP is formed by substrate level, so it is called a substrate level phosphorylation. So 2 ATP here, again 2 ATP here. So 4 ATP is producing. Out of the 4 ATP, 1 ATP is give back at this stage glucose to glucose 6-phosphate, and 1 ATP is utilized to fructose 6-phosphate to fructose 1,6-biphosphate. That means out of the 4 ATP formed, 2 ATP is payback. So this stage is known as the payoff stage. 1 to 5, that is known as the preparative phase, and this is the splitting phase, or it is known as a cleavage phase. And from here onward, ATP synthesizing, and out of the ATP formed, 2 ATP is given back. So this stage is known as the payoff stage. And finally 2 molecule pyruvic acid is producing, because here 1 glyceraldehyde phosphate come down, and dihydroxyacetone phosphate converting into glyceraldehyde phosphate. So from here onward, each and every molecule formed is 2 in number. So finally 2 pyruvic acid will form. Sorry, 2 pyruvic acid will form here. So that is the glycolysis. Then what is the significance of the glycolysis? The significance of the glycolysis: by the glycolysis, 2 molecule NADH is produced, and each NADH during the electron transport system converting into 3 phosphate group. So total 6 phosphate group will form later in the ETS. At the same time 4 ATP is produced, and this out of the 4 ATP, 2 ATP is given back, and 2 ATP is the net profit. Okay, so that is the significance of this type of the glycolysis. And another significance, this is the only respiratory mechanism in the case of the anaerobic organism. So in the anaerobic organism, by the glycolysis, they obtaining 2 ATP molecule. That 2 ATP is sufficient to live successfully, because the organism is the bacteria or the monosaccharide organisms that are requiring less energy. If more energy is there, that it cannot bear the cell, and the cell will burn. So in that organism, anaerobic organism, the reaction stopping at the pyruvate, 2 pyruvic acid will produce, and along with 2 ATP molecule and 2 NADH. This NADH is utilized for the fermentation reaction. In some microorganisms, fermentation reaction taken place. During the fermentation reaction, pyruvate undergoing reduction, and during the reduction, reducing agent is requiring. That reducing agent is the NADH. In the case of the aerobic organism, this NADH at the time of the ETS converting into ATP. So that is all about the glycolysis.